Bio 2. Continuing on with Chapter 9, we're going to continue to look at how invertebrates have evolved over time. And so we're going to start with what are the major trends in invertebrate evolution, so meaning what are the major kind of patterns that we can pick out and how they have evolved over time. So the appearance of each phylum in the fossil record represents the evolution of a successful and unique body plan. So what that means is that over time, as our animals have evolved, our invertebrates have become more specialized, and each one of our groups of invertebrates has a very unique body plan. It's kind of, again, it's changed over time. None of them are the same, but each one of them has been equally successful, and that's why they're still around today. And then, again, this next part, just kind of repeating myself, is that features of this body plan typically change over time, leading to the formation of many new traits. So remember, our sponges, again, being our very first group, they really didn't have a whole lot of specialized traits compared to talking about our last group with our arthropods, with our insects, with our mollusks, with our starfish, things like that. There's been a lot of change over time. So they all kind of had an original ancestor, but just over time they developed new traits, kind of became more advanced. And then we can look here and we can look exactly at the different traits that each one of our groups is going to have. As you can see, not a lot of specialization going on with their sponges, and then as the Cnidarians came along, we added more germ layers, we added body symmetry, and then still no cephalization, however. And then our flatworms, we finally did get a cephalization, so that at least got a concentration of nerve cells in one area we could call a brain, and our germ layers developed as well. And then we kind of had the protostome development as being observed, and we can see with annelids going up to mollusks and arthropods. Again, a lot of the major characteristics are the same, but it's just within those groups there's a lot of differences just in how they kind of carry out their everyday bodily functions. And then the echinoderms, kind of a different group there. They're very different because they're actually deuterostomes, so they do not have as much development there. And there we go, sorry about that. So then the deuterostomes, and so that's going to make them more closely related to our vertebrate animals, which we'll get into after this chapter. And so then kind of just going along with those trends of evolutions, so as larger and more complex animals evolved, specialized cells joined together to form things called tissues, which make up organs, which make up organ systems. Again, that's all review. And they work together to carry out complex functions. And flatworms, again, kind of just being an example of one of our earlier groups, they have simple organs for digestion, excretion, response, and reproduction. So they kind of have the most basic functions for those. Whereas the mollusks and the arthropods, remember, their organ systems are interrelated, so theirs are going to be a little more complex. And then for body symmetry, again, all invertebrates except for sponges will exhibit some type of body symmetry. So that's a kind of a big characteristic, just showing that they have a definite, distinct body plan. And remember, our two types of symmetry were Cnidarians and echinoderms. They had radial symmetry, where parts extend from the center of the body, and again, you can draw almost any number of imaginary planes as long as it goes through the center. It's going to divide them in half. And whereas our worms and our mollusks and our other arthropods, they exhibit bilateral symmetry, where they have a mirror left and right side. So basically you just have to draw one line going right through the middle in order to equally divide them, and again, bilateral meaning two sides. Then another big characteristic that is shown up throughout the invertebrates is cephalization, which again, another review word, is a concentration of sense organs and nerve cells in the front of the body. And then remember, we've talked about all this before, but invertebrates with cephalization can respond to the environment in more sophisticated ways than our simpler invertebrates can. So remember, sponges, they can't really do much, except some of them could secrete a toxin, whereas our insects, they can actually run away or decide to fight depending on what the situation is, because they have more cephalization, they're more kind of advanced and have better ways to respond to the environment. And then most worms and arthropods, our nerve cells are arranged in structures called ganglia. So again, just the worms, they're a little bit better off than the sponges are. However, they're not kind of up with the insects in the more complex area because they do not have a brain. So worms kind of fall in the middle. They can respond, but their response kind of isn't as detailed, isn't as complex as things like what an insect would be able to do. And then another big characteristic that's kind of shown up over time is segmentation. And over the course of evolution, different segments in invertebrates have often become specialized for specific functions. So we said this was a kind of first came up in our annelids, our segmented worms. They were the first group to really have this, and it allowed them to increase its size with minimal new genetic material. So that's kind of a big characteristic of segmentation as well. And so again, remember, just thinking back to all those different body structure, or all those different segments in earthworms, each segment kind of had a different function to help them carry out their various bodily functions. And then another big characteristic that we've talked about this many times is the formation of a column. And remember, we first came in with when we talk about flatworms, because again, it's just an extra body cavity. So there is no body cavity in the column, or inside of a flatworm. So they basically just have their digestive cavity where food comes in, and then it goes back out the same way. And it's just there, like kind of right in the middle of the worm. There's nothing really else to house any other internal organs, because that's what the function of the column is. And then we moved on and we had pseudocolumnates, meaning they had a fake column. So they did have a body cavity, but it wasn't quite there yet. It didn't have the definite structure made up that actually defines what a column is. So they kind of had a fake column, so it's close, but it's not quite there yet. But then most of our complex animals, again, they have a true column, and remember, it comes from tissue from one of the germ layers known as the mesoderm. And again, this should just all be a review. We've talked about this before, and again, now the body cavity is an extra place to house internal organs and things like that. And then our last thing that we talked about this early on, is embryo development. So in most invertebrates, the zygote divides to form a blastula, which we've talked about that before, which is simply a hollow ball of cells. And then we talked about, again, our difference between protostomes and deuterostomes, where the blastopore, or the opening of the blastula, will develop into a mouth, whereas deuterostomes, that blastopore forms an anus. And again, that's just clues to see which animals are more closely related based off the way they develop. So that's it for the first section of Chapter 29. Let me know if you have any questions.